We've now arrived at the cell membrane, the first structure every molecule encounters before entering or leaving the cell. Although it appears delicate, this membrane is one of the most sophisticated biological barriers found in nature. As we move across its surface, notice that the membrane is not a solid wall. Instead, it is flexible, dynamic, and constantly changing, allowing the cell to respond to its surroundings. At higher magnification, the membrane begins to reveal its true architecture. Millions of tiny lipid molecules are packed together in an organized arrangement, forming a stable yet flexible boundary. These lipid molecules organize themselves into two opposing layers, creating what we call the phospholipid bilayer. This unique structure forms the foundation of every animal cell membrane. Each phospholipid molecule has two distinct regions. One end is attracted to water, while the other avoids it. This simple property causes the molecules to assemble spontaneously into a double layer. Because both the inside and outside of the cell contain water, the phospholipids naturally orient themselves with their water-loving heads facing outward and their water-repelling tails hidden inside the membrane. The result is a remarkably stable barrier that separates the cell's internal environment from the external world while remaining flexible enough for movement, growth, and communication. But lipids alone cannot perform all the functions required for life. Embedded throughout the membrane are specialized proteins, each designed to perform a specific biological task. These membrane proteins vary greatly in shape and function. Some extend completely through the membrane, while others are attached only to one side, depending on the roles they perform. Many of these proteins act as channels or transporters, providing carefully controlled pathways for ions, nutrients, and other essential molecules to move across the membrane. Unlike an open doorway, these transport proteins are highly selective. They recognize only specific molecules, ensuring that the cell receives exactly what it needs while preventing unwanted substances from entering. This selective movement is known as selective permeability. It allows the membrane to regulate the passage of molecules rather than allowing everything to cross freely. Some molecules, such as oxygen and carbon dioxide, are small enough to diffuse directly through the lipid bilayer without assistance. Others require specialized transport proteins. Water, although abundant, also moves in a controlled manner. Its movement across selectively permeable membranes helps maintain the cell's volume and internal balance. Larger molecules, charged ions, and many nutrients cannot simply diffuse through the membrane. Instead, they rely on carrier proteins or channel proteins that guide them safely across. The membrane is also a communication center. Receptor proteins on its surface constantly monitor the external environment, detecting hormones, signaling molecules, and messages from neighboring cells. When these receptors detect the correct signal, they trigger a cascade of events inside the cell. In this way, cells communicate continuously, coordinating growth, metabolism, immunity, and countless other biological processes. Throughout every second of a cell's life, thousands of molecules are entering and leaving through these regulated pathways. This controlled exchange is essential for maintaining a stable internal environment, a condition known as homeostasis. As we approach one of these protein channels, imagine ourselves as a tiny molecule preparing to enter the cell. Rather than crossing randomly, we pass through a highly regulated gateway designed specifically for controlled transport. We've now crossed the membrane and entered the cell's interior. Ahead of us lies the cytoplasm, a remarkably active environment where organelles, proteins, and molecular machines work together to keep the cell alive. Our next journey begins there.